We look at flowing water, the heavy viscosity of oil, or the sudden flex of a muscle, and we see life in constant motion. But at a microscopic level, every single one of these processes occurs in a specific medium. 70 to 80% of all living cellular content is simply water. The remaining cellular structure is dominated by structured organic molecules, specifically lipids and proteins. Molecular shape and electrostatic charge dictate the behavior of everything happening inside a cell. To understand these abstract microscopic rules of chemistry, we can map them directly to the observable macroscopic physics you see every day across Uganda. Understanding how a whole organism functions requires looking first at how these foundational chemical parts interact. We start with a single molecule of water, H2O, one central oxygen atom bonded to two hydrogen atoms. Because oxygen pulls electrons more strongly than hydrogen, the sharing is unequal. This creates polarity, a partial negative charge on the oxygen and partial positive charges on the hydrogens. When multiple water molecules meet, the positive hydrogen of one is electrostatically attracted to the negative oxygen of another. This forms a hydrogen bond. Look at this image of vendors packing the stalls at a Wino market in Kampala. The market women are tightly packed, cooperating and moving together as a highly efficient system. Water molecules behave the same way. Their collective hydrogen bonds create cohesion and adhesion, which allows them to stick to each other and cling to surfaces, like pulling moisture up through the xylem vessels of a plant. This network of polar charges creates an environment that easily dissolves other ionic and polar substances, making water the universal solvent for cellular metabolic reactions. If water is the medium, lipids are the insulators. They are a diverse group of nonpolar, hydrophobic organic molecules made of carbon, hydrogen, and oxygen. A simple lipid, or triglyceride, forms through esterification. One glycerol molecule joins with 3 fatty acid chains, releasing 3 molecules of water. If these fatty acid chains have 0 carbon-carbon double bonds, they remain completely straight. This allows them to pack together tightly into a dense, solid structure known as a saturated fat. But add a carbon-to-carbon double bond, and the chain develops a sharp physical kink. This prevents the molecules from stacking neatly, keeping the substance fluid, like the unsaturated sunflower oil used in cooking. This image shows a local example, ghee. Because its molecular chains are saturated and tightly packed, the substance remains solid at room temperature. A single microscopic variation, the addition of a double bond, dictates the exact physical state of the lipid. To build a cellular boundary, the simple lipid requires a structural modification. The 3rd fatty acid chain is replaced with a phosphate group, creating a conjugated phospholipid. The molecule is now amphipathic. It has 2 conflicting sides, a hydrophilic, water-loving phosphate head, and 2 hydrophobic, water-fearing lipid tails. When thousands of these molecules are exposed to water, they spontaneously snap into a tight 2-layered sheet, called a bilayer. We can see this dynamic in this aerial view of Lake Victoria's shoreline. Boundaries are naturally drawn by opposing environments. The hydrophilic heads of the bilayer act exactly like the sandy shore facing the open water of the lake. Meanwhile, the hydrophobic lipid tails mirror the dense, dry inland area, pointing strictly inward to avoid the water entirely. This amphipathic structure spontaneously constructs the plasma membrane, creating the stable barrier necessary to separate the interior of a cell from its environment. With the liquid environment established and the lipid boundaries drawn, the cell needs machinery to do the work. That is the role of proteins, built from repeating units called amino acids. Every amino acid has a central alpha carbon hub. Attached to this are an amino group, a carboxyl group, a hydrogen atom, and a variable R group that gives each amino acid its unique chemical properties. The carboxyl group of one amino acid joins the amino group of another through a condensation reaction. They lose a molecule of water and lock together with a strong covalent peptide bond. When many amino acids join, they form a specific linear sequence dictated by the organism's genetic code. This is the primary structure. Because this exact sequence dictates the final shape, Altering just one amino acid breaks the protein's utility. This microscopic image of blood cells shows the consequence of that error. Among the normal round cells are crescent, sickle-shaped cells, the direct physical result of a single incorrect amino acid in human hemoglobin. The primary structure is the raw material. A linear sequence must be properly folded before it can perform biological labor. Think of this primary sequence like the flat individual reeds in the hands of this weaver. They are unbent raw materials waiting to be shaped. The local folding of the chain into alpha helices or beta-pleated sheets is the secondary structure, held in place by hydrogen bonds. The overall shape then folds further into a three-dimensional form. This is the tertiary structure, stabilized by interactions between the R groups, much like the intricate weaving and precise knots of a finished Kagezi basket. This folding yields two main types: long, structurally strong fibrous proteins, or compact, biochemically active globular proteins, like enzymes. The exact three-dimensional shape creates the active site, a pocket perfectly contoured to grab molecules and execute a specific metabolic job. If the surrounding temperature spikes or the pH changes, those stabilizing bonds break. The structure unspools back into a loose thread, a process called denaturation. If the bonds break, the structure unravels. Just like an unraveled basket can no longer carry grain, a denatured protein loses all function. These three independent models, the polarity of water, the saturation of lipids, and the folding of proteins, do not exist in isolation. They synergize into a single working engine. A folded protein slots into the phospholipid boundary, functioning within a liquid environment. Water acts as the dynamic medium that makes all transport and chemical reactions possible. Lipids serve as the hydrophobic architects, compartmentalizing the space and protecting the system. Proteins operate as the precision-folded engines, executing the exact labor required to keep the cell alive. Biological life operates through electrostatic architecture, where every movement and reaction is a direct consequence of molecular structure.